So welcome to your next section in genetics. So this is section 1.3 in your textbook, and this is part 1. So we're now moving on to mechanisms for diversity of life. So this is mutation and its impact on evolution. So when we think of mutations, we think of things in movies or things going horribly wrong. But in reality, most mutations are not as horrible as you'd expect and are important for evolution. What we're going to do on this slide is look at a couple of picture examples of mutations, and I want you to think about whether or not they are real or fake. So our first example here, take a moment, think. By the way, this is a vegetable called okra. So this is indeed a real mutation. So changing color from green to yellow is the mutation. Next one here, real or fake. So this is a lamb, and looking at this particular lamb, something seems a little bit off. So we can see 5 legs and 6 feet. So this is a real mutation, and it is a mutation in the gene involved in growth of limbs. Next one, real or fake. So here we've got a dog with a rooster head. So this is not what we're talking about when we discuss mutations. This is an example of Photoshop. Next one, real or fake. So again, fake example of a mutation. Peter Parker is bit by a spider that gives him the powers that Spider-Man has. So unfortunately, this is not a real mutation. So I'm just going to skip through these slides really quickly. They're just related to your assigned readings in the notes. So section 1.3, mutations and genetic diversity. Question for you is, what if something goes wrong during protein synthesis, which is the translation phase? So when you are going from DNA to RNA, what happens if it didn't get copied correctly, is another way to put this question. So the answer is, if translation is altered, the protein product will be different. So if DNA isn't copied properly to RNA, then the protein made from it has an incorrect set of bases. The protein is made from an incorrect set of bases. So protein function depends on its shape. If the shape changes, its function changes too. So protein function depends entirely on the shape of the protein. So remember the lock and key analogy. If you were to change the shape, the function will change as well. So mutation can change in the base sequence of DNA. When bases, the letters change, the instructions used to make the protein also change. Different amino acids will be used, resulting in a different protein. This may alter the protein shape and also change or break protein function. So the effects of different mutations, or different types of mutations. Mutations are often a bad thing because the protein does not work and your body needs it to function. These are known as negative mutations, and they decrease survival rates. So for example, a mutated gene results in sickle cell anemia, which is misshapen red blood cells that don't carry oxygen well or efficiently. So normally you can see the pictures that's drawn in here. Blood cells are quite circular, they're kind of plump, and in sickle cell they become kind of like a moon or sickle shape. Sickle cells, as it says, they don't carry oxygen well, so that's a bad thing. But that being said, if you live in a malaria zone, having this mutation isn't actually a bad thing because it makes you less likely to die from malaria. So although it's still a serious mutation, it has a potential benefit to it. This isn't the case for all negative mutations, but in this case, it does have that. Another example is cystic fibrosis. So more than 1,300 different mutations in one gene cause cystic fibrosis, which is basically a disease that involves a buildup of mucus as well as some other tissue differences in the lungs. Cystic fibrosis, essentially you feel like you're drowning, and there's a whole bunch of mutations that can possibly lead to cystic fibrosis. So let's talk about these two in a little bit more detail. So here you can see a healthy blood cell versus a sickle cell anemia red blood cell. So sickle cell is sort of like banana-shaped, moon-shaped, sickle shape instead of plump and round. The round cells will easily flow through your red blood cells. So you see that in the unrestricted blood flow here versus the sickle cells, which can end up blocking the flow because they get caught on each other, which is kind of what you're seeing right here. So this obviously causes problems in blood clots, that sort of thing. So not good. Cystic fibrosis, a healthy trachea is on the top. So here, you can see it's quite wide and open, lots of air can pass through. This is referring to your windpipe. Versus if you have cystic fibrosis, you have a thickened muscles which narrow the pathway. And in addition to that, there's also an increased mucus and swelling and a bunch of other things that make it a lot harder for an individual with cystic fibrosis to breathe. So sometimes mutations can be a good thing, i.e. the mutated protein works better than the normal protein or has a new beneficial function. These are known as positive mutations and they increase survival rate. For example, a mutated gene that exists where a protein basically makes individuals resistant to HIV infection. So these people cannot contract HIV and therefore AIDS. So this is pretty incredible since people have worked hard over the years since the AIDS crisis to extend the lifespan of people affected by HIV. But we don't actually have a successful vaccine yet to prevent acquiring the virus. So we can only treat the virus, not cure it. So you live with HIV for your entire life, essentially. So yeah, there are people who have this mutated gene that makes a protein that makes it so that you can't get HIV or AIDS. Some plants have mutated genes that code for proteins that make them resistant to chemical herbicides. So in the bottom example here, obviously being resistant to herbicides is bad for, say, a weed that you're trying to kill. So from your perspective, it's bad, but for the plant, it's good because it's increasing their survival. So that one plant is resistant, it grows up, it produces seeds, which then eventually lead to a new population and potential evolution down the line. So in addition to positive and negative mutations, we also have neutral mutations. So sometimes mutations have no effect because they occur in a part of the DNA that is not used for protein synthesis, or the mutated protein is not required for survival. So these are known as neutral mutations and have no effect on the organism. So remember that genes are on specific segments of the DNA and that not all of the DNA codes for genes. So if a mutation occurs between the genes, it may not matter or have an effect. So for example, mutation in a gene that codes for fur color in the spirit bear. Although white, the survival rate of the organism is not changed. So for the spirit bear, instead of being a black bear, the spirit bear doesn't have the ability to make the proteins which result in black fur color, but this doesn't affect its survival rate. A big reason for this is that the bears are at the top of the food chain, obviously, versus something lower down in the food chain. They are omnivores, so their risk for death is pretty low. As a potential benefit, although they're really not sure about this, the spirit bear may actually be better at catching fish because of how the sun reflects off of the water. It blends them in a bit from the fish perspective. In this case, it is considered a neutral mutation. It's just changing the color. It's not affecting its survival, so there's no real downside or upside. So consider the following question. Would you expect a positive, negative, or neutral mutation to lead to genetic diversity? And why do you think that is? So I'd like you to take a moment to think about this, pause here, and basically think about what's going to result in more genetic diversity. Negative obviously is bad, so it will generally kill off the organism, so it's not likely to be passed efficiently. So negative is out. Positive and neutral can both create diversity. But positive is more likely to cause a whole species to work towards that new thing, where neutral will stick around but doesn't really benefit positively or negatively or impact the species. So think about something like eye color, for example. It is a neutral mutation. Originally, brown eyes likely evolved to shield against UV light through an increased amount of melanin produced. But in a world where we now have things like sunglasses, your eye color really doesn't matter. So the impact on the population as a whole is low. It doesn't really impact, but continues on and is passed on to future generations. So therefore, positive is most likely to impact genetic diversity. So section 2.1, there are two types of mutations, gene mutations and chromosomal mutations. So let's talk about those a little bit more. So a gene mutation is a mutation that occurs within a gene at some point along the chromosome. This mutation is only a change of one or a few bases or the letters of DNA. It usually only affects one gene and therefore can have less impact than larger chromosomal mutations. So recall in protein synthesis that DNA in the nucleus codes for RNA, which then goes into the cytoplasm and into the ribosomes where a protein is produced. So this is kind of an overview of that whole process. So DNA in the nucleus, RNA in the cytoplasm, protein in the ribosomes, which are also in the cytoplasm. So you've got a code here, ACT, GCT, which then gets coded into the complementary RNA, which then codes for some kind of amino acid change. So if the sequence of DNA is changed by a mutation, then the RNA sequence copied from that DNA will be different, and this will code for a different amino acid, which results in a different protein. So this is called a gene mutation. So for example, the spirit bear is white because the gene that codes for fur color, which is dark brown or black, is mutated and no longer works. So there's our example. Mutations can lead to genetic diversity. Individuals within a population have different traits. And if there is a selecting factor, the development of a new species. Example, if a mutation causes a fish to have spines, so spikes basically, and a new predator is introduced to the area, but it cannot eat the fish with the spines, this may select for evolution of a new spiny fish species. So there's a little drawing of it in the bottom there. So if predators can't eat the spiny fish, then they will be able to breed and pass on that spiny gene, meaning the trait will be selected for and could eventually result in evolution of a whole new population of spiny fish. So there are three main ways that gene mutations can occur. The first one we call substitution. So for example, in the string of DNA bases, so A, C, T, G, C, A, if this C here is changed to an A, then this could result in an entirely different amino acid sequence, therefore changing the protein, etc. Deletion is when we lose a base altogether. So looking at that same sequence, all of a sudden we've lost this T. So everything is shifted to the left. So this is called a deletion. These, by the way, when you look them up, may have slightly different terminology online. Okay, so just so you are aware. Okay, and then our last one is the addition, or also could be called an insertion mutation. So essentially, for our example here, we're adding another A in. So that's the space here. So it basically shifts everything down further, again, kind of messing with the potential amino acid sequence for the protein. Okay, so deletion or addition mutations can lead to what is called a frame shift. These are the most harmful because all the code afterward is changed. It will give a different protein or stop coding altogether if it codes for a stop codon. So the protein shape could change entirely or just not be formed because it stops at a stop codon. So again, this will all impact its function. So at the top there's an example of a sentence, the cat ate the hat. It's using three-letter words because it's supposed to relate back to DNA. So if we all of a sudden were to delete that H in D, the entire sentence no longer makes sense. So this is kind of give you a visual of what a frame shift mutation does. The extent of damage caused by a gene mutation also depends on where the mutation occurs. If, for example, the mutation occurred at the beginning of a gene sequence and caused a frame shift, the damage would be much greater than a mutation that occurred at the end of a gene sequence. So it completely depends on where that mutation occurs, determines how many amino acids could be affected. Okay, so determine the extent of the damage caused by each of the following scenarios. So I'll help you through the first one, but essentially I want you to think up of an example of what this would look like if the change was to occur, and then think about what's the extent of the damage. So for number one scenario, deletion occurs in the start codon. So that AUG is no longer an AUG. Without the start codon, there can be no translation, therefore no protein can be made. So this is a high extent of damage. The protein would not be produced. I would say instead of the gene would not be produced. I mean, I guess it's kind of like the gene wouldn't be produced because without the start codon, it's not really a gene, but really ultimately it's the protein that's not produced. For B, an addition occurs near the beginning of a gene. So think instead of it being in the start codon, maybe it's like here. So what would this look like and how damaging would it be? What's the extent of the damage? C, a substitution occurs at the beginning of the gene, again in that same kind of ATT region. You don't have to use that exact string, by the way, you can write your own. But what would that look like? What's an example of it? What's the extent of the damage? A deletion occurs at the end of the gene. What would that look like? How does it affect it? And an addition occurs in the non-coded part of DNA. So this is the part that doesn't go on to make a gene or to go on to make a protein. It's not within the gene. What would this look like and how damaging would it be? So we'll go over this later on, not in this video, but take a moment to try to answer these questions. So on page 56 of your notes, there's a quick exercise to work through. This is an example of what it looks like. So 4-2A, identify the mutation. So what it would like you to do is to copy a DNA sequence into your notebook, separate the bases into groups of 3 to make it easier to work with, and then figure out if these examples are substitution, deletion or deletion/lost, or addition of a base in step 3. And then there's a few discussion questions that go along with it, okay? So this is the end of the topic for now. We'll see you back in our next video.